Okay, so the markers are going to start correcting today. So as soon as everything is done, there will be an announcement on Blackboard. The marks will be up, and you'll be able to go pick up your exam. And we'll go through the same process where you'll be able to send in a request for remarking by placing an argument at the back. And it should be an argument. It should be the marking scheme says this, my answer says that. There's 2 marks missing. There's 1 mark missing. And it shouldn't be, I really think that this should have been on the marking scheme. And it shouldn't be, I really think that my answer kind of means what's sort of laid out on the marking scheme. That's not an argument. That's a plea for pity. Okay? So make sure that you go over your exam, because some mistakes do happen. You know, it happens. You're correcting a whole stack of 1,400 exams. You can slip up on 1 or 2 of them. That's why you have the ability to send in your exam for remark, but it should be for a real remark and not just trying to get our sympathy. Okie dokie. So before the midterm we left off, we had started talking about transcription, and here we are. We have transcription going on between prokaryotes and eukaryotes. So we've seen a few differences. Now we're left with a mature RNA transcript in prokaryotes and an immature transcript in eukaryotes. So we need to mature that transcript. So we talked a little bit about the poly A tail. We talked a little bit about the 5' cap. And we left off with the idea that there are some coding and non-coding regions, the exons and the introns. And we have to find a way to make sure that we can get rid of the introns so that we have only a coding sequence in order to get into translation. Okay? We don't want to translate non-coding regions. We're going to have improper protein synthesis. Okay? So we need to clean that up. So let's make sure that everybody's wide awake. Which of these 4 statements does not correspond to a difference between transcription in prokaryotes versus eukaryotes? So which one of these is not something that's different between prokaryotes and eukaryotes? Bless you. Who needs more time? All set? All right, let's see how you did. Aha! All right, find a buddy, read your notes, go on the internet, find the right answer. tell you what, it's early for me too. All right. How come we've got such a dissension this morning? And now you're all shifting your answers. So what's going on? Let's go over these possible choices. We're looking for something that's not different between prokaryotes and eukaryotes, okay? We're looking for something that's the same. We looked at gene organization, and we looked at how the sequence within the promoter is organized between eukaryotes and prokaryotes. Is that the same? No. Did we talk about a Tatabox when we talked about prokaryotes? Did we talk about activators and repressors and initiating a transcription initiation complex? No. So that is different. So A is incorrect. We talked about transcription itself. So running through DNA using a RNA polymerase and transcribing that information onto a new strand of mRNA nucleotides. Was that done differently between prokaryotes and eukaryotes? Did we have an enzyme, a different enzyme? No, it's the same polymerase. Okay? What recruits it and how it's organized is a little bit different, but it's the same polymerase. But what you obtain is different. In prokaryotes, you get right away a mature transcript, whereas in eukaryotes, you get an immature pre-messenger RNA that you are going to have to mature. That's post-transcription. Transcription has already been done when you need to go and clean up your transcript, okay? And the initiation does not occur the same way in prokaryotes versus eukaryotes. The protein involved, the sequence, all of the complex formation to trigger RNA polymerase II from reading that strand is different between eukaryotes and prokaryotes, okay? So whoever's got B, kudos on a Monday morning. All right. So let's look at this pre-messenger RNA transcript, okay? So we talked about the poly-A tail at the 3' end. Who can tell me a little bit about the poly-A tail? Poly-A tail? Anyone? Going once? What's it made of? Adenine. Okay. What else? How many? 1, 2. Long. Okay. How long is long?
50 to 200.
Yeah, 50 to 200. Okay. So it's quite a long tail. What's it good for? What's it good for? What is its main claim to fame? Why do we need it?
Like generally speaking, it helps the mRNA get out of the nucleus.
Yeah, it's not just generally speaking, it's a requirement. Without it, that mRNA is not going anywhere. Okay? There's another important reason why we have that poly-A tail. Yes?
If like an enzyme in the cytoplasm degrades it, it degrades the tail first as opposed to degrading the actual messenger.
But not in the cytoplasm. You're still in the nucleus. Okay? So nucleases in the nucleus, if that poly-A tail is there, then it will prevent the mRNA from being degraded. And that will allow it to get out of the nucleus. Okay? What about the 5' cap? What is it made of and when? Okay? When? During, before, or after transcription? Who said after? Okay. Who says before? Okay, good. Who says during? Not enough of you. It happens during transcription. Okay. Once the polymerase starts transcribing, once it's about 40 nucleotides into transcription, that's when the 5' cap is being added. Okay? Okay. Is the 5' cap and the poly-A tail part of the coding region? Is that going to be translated into a protein? No. Okay. Good stuff. So now we're left with all of these exons and introns, and we're stuck with that, and we've got to clean it up. Okay? So we need to get rid of the non-coding sequence, the introns, and we're going to do that by splicing. Okay? This is the essential, the core of RNA maturation, and it's called splicing, and it's accomplished by a structure that's called a spliceosome. A spliceosome is essentially a structure that's made up of protein and small nuclear RNAs that assemble together, and they're able to recognize specific sequences on your transcript. Which sequences? Well, they are able to recognize the beginning and the end of all of your introns, and they bind to them. Okay? So they are made of small nuclear RNAs, so they're able to be complementary to your RNA transcript. And there are proteins that come along for the ride, because they're going to be needed to do the actual cleavage, the actual cutting, and also to be able to reattach the exons. Okay? When you cut out your intron, well, the adjoining exons on both sides of the intron you're trying to remove need to be reattached. They need to be reconnected. Otherwise, you're left with 2 pieces, or 3 pieces, or 4 pieces of RNA. That's not what you want. You want to have one complete coding sequence. Okay? So these proteins are involved in the cutting and reassembly of the exons. Okay? So small nuclear RNAs and small proteins will allow to recognize sequences at the beginning and the end of your introns. The protein that come along within the spliceosome complex, once those pieces are attached, so your complementary strands now form a double strand, that serves as a signal. Your proteases that are part of this complex will come in and cleave that area, removing the intron. The intron is excised, so it's no longer part of your transcript. It's going to be degraded and whatnot. But the other proteins are there to make sure that they can join these 2 portions together. So you're left with a 5' cap, all of your necessary exons, all the way to the end at the poly A tail, with no longer any intron or non-coding sequences. Okay? So let's take a typical pre-messenger transcript. Okay? That can be, on average, about 27,000 nucleotides. Okay? And once you've removed all of the non-coding introns, you're going to be left, on average, with about 1,200 nucleotides. And when that goes on to translation, you're going to have a protein that's about 400 amino acids. This is just an average. There are proteins that are much smaller, and there are proteins that are much larger. Okay? So you need a transcript to be long enough in order to have the number of amino acids that your protein requires. But it's just to give you an appreciation of how much of that sequence in the pre-mRNA transcript, how much of those introns represent in terms of size, in terms of number of nucleotides. We're not talking about getting rid of just about 10 or 15 nucleotides. We're talking about getting rid of most of the transcript. Okay? So we transcribe, it ends up a long transcript, and we're going to clean it up so that we have just what we need. Okay? Okay. Now, let's talk a little bit about regulation, about these different events. Okay? So we just talked about splicing. What if during transcription, you remember that RNA polymerase II goes faster than DNA polymerase III, it doesn't back up, it doesn't proofread, and it doesn't make any corrections. So there are mistakes that are going to be left behind in your transcript. What if that mistake, or those mistakes, depending on how many there are, what if they're just smack dab where the spliceosome would have recognized that portion as being an intron or a non-coding region. What do you think happens? And just scroll down so that choice D is also obvious. Who needs more time? Okay. All right, let's see how you did. Oh. Hello. Okay. So, no splicing. Okay. Normally at the other introns, but only part of intron 1 will be excised, whereas it'll be degraded. Oh, interesting. Okay. Let's go back just for the sake of it. Okay. When your spliceosome, when the ribonuclear portions within the spliceosome come in, they recognize specific sequences at the beginning and at the end of the intron. Both need to have matched. Otherwise, cleavage can't occur properly, and you can't reattach. You'll be left with a hanging portion. Okay? So if you have a mismatch, an error within the sequence, right where that recognition sequence is located, then those ribonuclear proteins won't match and won't recognize properly. So the other proteins involved in the spliceosome won't be able to achieve the cut properly. But that mistake can occur here for intron 1. But the rest of the sequence is intact. So all of the other introns that need to be spliced will be spliced correctly if there aren't any mismatches there as well. Okay? So the correct answer here was B, where only the other introns will be spliced correctly, but not the first one. So you'll be left with a non-coding portion left behind in your transcript. So what consequence will that have? What impact will that have?
Protein that's necessarily, that may be necessary won't be coded for.
Yeah, so if it moves on to translation, then you're going to translate that non-coding sequence, and you're going to get a protein that's inefficient. Okay? That's assuming that the entire transcript gets translated, because within that intron, there might be a sequence that corresponds to the signal that tells the ribosome to start transcribing. We would have to look at the entire sequence to be able to answer definitely what the impact will be. But certainly there is going to be an impact on that protein. Okay? Okay. So this brings us back to our splicing. Do we need all of the exons that are available? We need to cut out all of the introns. They're non-coding, so we need to cut them all out. But do we need all of the exons? Why not? They're coding, right? So why don't we need all of them? Do tell.
Because we don't necessarily need the gene at the time of that gene to be expressed all the time.
Well, this is part of the gene. When you say the gene for insulin, there's so many exons and so many introns. You're going to get rid of the introns, and now you've got all of your exons, and you've got insulin that's able to be translated. But do you really need all of those exons? That's the question. Do you need all the exons, you think? Okay, so I see some shy yeses and some even shyer no's. Okay, so we talked about different isoforms of protein, right? Remember when we talked about proteins, we talked about members of a large family that are all part of the same umbrella type of protein. For example, we talked about phospholipase C, and there was one that particularly liked to interact with tyrosine kinases, and another one that particularly liked to interact with G-protein coupled receptors. They're part of the phospholipase C family, but they're not exactly identical in sequence and in structure, and therefore in cellular location. How did that happen? It's the same gene. So how come we've got different proteins that do similar tasks, that are part of the same family? How did we accomplish that within the sequence of DNA? How can we get different flavors of different protein? Any suggestions? Come on, give it a shot. What do you think? No idea? No? I thought you had one and no. Okay. Yeah?
I'm guessing like the different exons will add different characteristics that are needed for some proteins and not needed for others.
Ah, very good. You had something? Same thing. Okay. Anything else? So different exons could lead to giving different results or different characteristics to different proteins. This is what we're talking about, right? Isoforms of the same protein give them different flavors. Okay, so how do you think we can accomplish that? What mechanism could we use to make sure that we still have a complete protein, but they have slight differences, and yet are part of the same family of proteins? So they could have different cellular locations, maybe they could have different affinities, slight differences in the ligand binding domain, or maybe just be expressed more favorably in one cell type versus another. How do we get that? We have one gene. Yeah?
Maybe changing orders of exons?
Ah, changing the order of exons. Okay. That's a bit of a challenge to reorganize them, but I like it, I like it. What else could you do? Yeah? Change the length of the exon, yeah?
Start and stop region.
Okay.
A little sooner and something a little later.
Okay, so stop a little sooner and start a little earlier or later. Change the length of the exon, yeah? Turn different ones on and different ones off. Okay, I like that. Yeah? Well, you've already got a protein there. Yeah, okay. So absolutely, hang on to that when we get into translation. Now we're trying to do it at the transcription level, okay, before we actually go out and meet our ribosome. But you're correct. You're just one lecture early. Okay. Turn certain exons on, use different lengths, maybe change the order. Think about what we just talked about. When we remove introns, right? When we splice our transcript. Look at the structure. Think about those different suggestions, which are all very good. Now try and figure out how the cell could accomplish that. How could the cell accomplish changing the length of the transcript or using different ones by turning some on or off? Yeah?
In the diagram, you see either the start codon or stop codon, and whatever's in between. Once you recognize an intron, any exons between those introns, it splices it up.
Aha. So what if instead of splicing here, you were to splice here and here, or here and here? Is that what you're driving at? Absolutely. Very good. So that will result in that exon being removed, and you would have a much shorter transcript. Okay? So genes that lead to multiple isoforms or multiple members of the same family of protein will have that built in. They will have some sequences that you can or cannot use. And that leads us to alternative splicing. Okay? So within the gene, you'll prepare your transcript. And at the stage of splicing, when you're getting rid of introns, you will need to get rid of all of the introns. But you can use that opportunity to remove some exons as well. And that will lead you to different flavors of the same protein. You have here an example of a pre-mRNA transcript with all of the possible exons, numbered 1 to 12. And this is the transcript for tropomyosin. Tropomyosin we talked about when we talked about muscle contractions. We talked about it. It's the protein that wraps around actin microfilaments. And it's the one regulating the binding sites between actin and myosin. You remember that? Okay. So tropomyosin is present in skeletal muscles and in smooth muscles. But the shape of tropomyosin, the way it's physically organized as a protein, is different in these two cell types. But it's the same protein. It does the same job, but it does it a little bit differently. Okay? So how can you accomplish that? Well, when the gene is transcribed in each of those cell types, the way the transcript will be spliced is different. In smooth muscle cells, you're going to splice it in a way that you're going to keep exons 1, 2, 4, 5, 6, 7, 8, 9, and 12. Whereas in skeletal muscles, when the gene is transcribed, the spliceosome will go and mature that transcript, saving only exons 1, 3, 4, 5, 6, 7, 8, 9, 10, and 11. Okay? So there are certain exons that are identical, which means that when that transcript will be translated to protein, that sequence will be identical. That portion of the protein will be identical. But there will be some differences. And if you look carefully, the middle part is exactly the same, and the edges are different. Okay? When we look at translation, we're going to see that the order of the exons correspond to an area of the protein. Because when you transcribe your gene, you have your nucleotides in order, and when you're going to translate them, that's going to lead you to an ordered sequence of amino acids, right? So the extremity of your protein is at the extremity of your sequence, okay? You don't reorganize that afterwards, okay? So those proteins are very similar, but they have differences in a few exons, but the core of the protein is the same. But it means that they carry similar roles, but certain particularities depending on the tissue type or the cell type that they're expressed in. You had a question? So within the spliceosome, the different sequences that are able to recognize that, within the complex, the proteins that assemble it, that's where specificity will occur. Okay? So different cell types will produce different proteins and build their spliceosome differently. So in the example with tropomyosin, the spliceosome, the mechanism is the same in skeletal muscle and in smooth muscle cells, but the actual sequence and the recognition parts and the enzymes that are involved will be different. They work the same way, but they're looking for different sequences, which means that when tropomyosin is transcribed, you're going to obtain a different sequence of splicing. Okay? So, and that's true for all of the different proteins, right? Okay. Yeah?
Because we're seeing that like the middle ones are the same, right?
Yeah.
But only the outside ones are different. Does that mean the middle ones are the key elements for determining what the protein will actually be?
Yeah. Well, the middle, we're actually going to see how exons are related to domains in the protein, right? So we talked about ligand domain bindings and we talked about catalytic domains and we talked about transmembrane domains. Well, they correspond to these different exons, okay? Different exons will lead to a sequence of amino acids, and together they'll have a specific job because they're present in the protein at a certain area, okay? So we're going to get to that when we talk about translation, of course, but yeah, you're absolutely correct. Okay. So now we need to talk about how you regulate the expression of different genes. And again, this is where there's going to be a difference between prokaryotes and eukaryotes. So let's start with prokaryotes. Okay? In your book, you're going to see three examples of gene regulation in prokaryotes. Okay, we're going to talk about the lac operon this morning, and you're going to see in your book there's the cyclic AMP and the tryptophan operon, I think. Okay, so read all of them, but for the exam, you're responsible for the lac operon. Okay, but do read up on the other ones as well. They'll help you understand the mechanism and how it works. Okay, okay. So what is an operon? An operon is the name given to the structure within a prokaryotic gene that allows to control the expression of that operon. And the operon is usually an arrangement of more than one genes that need to be transcribed together. Okay? So in this example, in the lac operon example, this is 3 specific genes that when they are transcribed and translated, they lead to 3 enzymes that are essential for the prokaryotic cell to digest lactose. Okay? So it makes sense that whenever that cell is in the presence of lactose, it's going to need to digest lactose. To do that, it needs those 3 enzymes. So it makes sense that the regulation of those 3 genes is coordinated. That you should produce those 3 enzymes at the same time because you're going to need all 3 of them to accomplish digestion of lactose. So instead of regulating all 3 separately, the genome is organized in such a way that there's one switch that allows to turn on transcription of all those 3 genes, and that same switch allows to turn transcription off. Okay? That's what an operon does. One switch, and it allows you to organize or to regulate the expression of more than one gene. Okay? So here we have our regulatory gene. This is the gene that controls the operon. Okay? Then our operon. We have our promoter, the area where the RNA polymerase will need to come in once it recognizes its sigma factors, binds to that area, and initiate transcription. But right next to that promoter, before the transcription start site, there's what's called an operator. And an operator is a sequence of nucleotides right after the promoter, before the start of transcription, that's able to be recognized and attached to by protein. And these protein are called repressors. And the repressors are produced by the regulatory gene. So when the regulatory gene is transcribed, it will lead to a repressor. That repressor can come in and recognize the operator. When it does, the result is very specific and very tight binding, which means that the polymerase can come in and recognize the promoter, but it can't move forward. It can't go anywhere. You've essentially put up a roadblock on that DNA sequence. Okay? So there's a barrier, which means that your polymerase won't be able to move forward. It won't be able to transcribe either of the genes that are under the control of that specific operator. Okay? You can have many different operators. Okay? The idea is to have a switch to control how and when different genes will be expressed when they need to be expressed at the same time. Okay? Okay, let's look at it in the two conditions that can occur for that cell. The cell can either be in the presence of lactose or in the absence of lactose. Okay? So when there is no lactose, the cell doesn't need to process lactose, doesn't need to digest it, so those enzymes are useless. So we're going to turn off transcription of those enzymes. How are we going to do that? We have our regulatory gene that has been transcribed. In this case, it's LAC1. So that regulatory gene will have a given name for each type of operator you're looking at. In the lactose operator, it's called LAC1. The LAC repressor is translated in active, and it's going to recognize the specific sequence right after the promoter called the operator. Latch onto it and bind specifically. You now have a gate. You have a roadblock. RNA polymerase, it can't fit anymore. You have a roadblock. So it can't fit, can't attach, can't recognize to the promoter, therefore it can't transcribe. Okay? So those three genes that are under the control of that operator will not be transcribed. There won't be any translation, and you won't have any beta-galactosidase, any permease, or any transacetylase that would be required for that cell to get rid of lactose, to process lactose. Okay? Now the cell is in the presence of lactose, so it needs to deal with that lactose. So we need to get rid of that roadblock because we're going to need to initiate a little bit of transcription so that we can obtain these three enzymes. Okay? And thankfully, the three genes are next to each other, so we're going to be able to initiate transcription once, get all those three genes transcribed, and get one long mRNA transcript that's going to be able to produce all three of these enzymes. Okay? So how do we do that? Well, when there is no lactose, we have our repressor that's being produced. Okay? And it binds to the gene. It binds to DNA. When you have lactose, lactose will be present, and it will be converted in the cell to allolactose. And allolactose is what induces your operon. It's what turns it on. It's how you detect that you need to get rid of the roadblock. And your repressor protein that was bound to the operator, well, when there's that allolactose present, it has much stronger affinity to that than to DNA. So it's going to detach from the operator, and it's going to bind to your allolactose. So now the operator site on DNA is free. Aha. So now your polymerase can bind to the promoter. There's no longer any roadblock. And as long as there's lactose, it's going to keep your repressor busy. So you're free to transcribe. Polymerase keeps going, and it transcribes your three genes, getting your transcript out. Translation will go on, and your enzymes required to process lactose are now present enough for you to be able to get rid of your lactose and use it. Okay? So, is this fail-safe? There's no mistakes, it's always 100% efficient? You're right. It's kind of like, most of the time it works. Every once in a while, this interaction can sever itself. The relationship can stop for a little while. So the operator can drop the repressor. It can be because of DNA movement. It could be because of unspecific detachment. Many reasons. But it doesn't happen very often. What happens if it does? So there's no lactose, and you've dropped your repressor. So what happens to those genes? Well, if you drop the repressor, the genes are going to be transcribed, right? But very few of them will. Like there's only like that short period of time where you drop your repressor, the transcription will occur. So that means that you're going to have low levels of these enzymes, even if there is no lactose present. Okay? Very few, very low levels, little bit of these enzymes. And that's important. Why? Why is that important? Because initially, when the lactose becomes present in the medium, well, you're going to need to get it inside the cell. Is the membrane permeable to lactose? What's lactose? What kind of macromolecule does it look like? Yeah, it's a small sugar. It's carbohydrate. Okay? Membranes aren't very permeable to carbohydrates. So how are we going to get it inside the cell? What type of membrane transport will allow to get it inside the cell? Facilitated diffusion. So we're going to need a channel, a transporter of some sort. Oh, look at that. We have a permease here. Okay? So the fact that our operon is not 100% efficient is what allows us a little bit of a safeguard to make sure that when that sugar becomes present in the media, we can actually get the reaction going. Okay? And the same is true for the enzymes that start getting things done. The permease allows us to get lactose in. The beta-galactosidase allows us to convert it to allolactose so that we can get the repressor out of the way. Okay? So nice and pretty well organized for small prokaryotic organism, don't you think? Yeah? Okay. Questions on this? Yeah? No? Yep. Well, only this is prokaryotes, right? So it's different for eukaryotes. But if there were to be prokaryotes that would be lactose intolerant, this would be where the issue rises. Yeah? Good question, though. Anything else? Yeah?
There's something about direct reaction of RNA monomers, or does it just bind to what the promoter is?
Well, okay, so this speaks to initiation of transcription. So you need those sigma factors that we talked about previous lecture, okay? But when the repressor is there, even if the sigma factors are there to recruit it, there's just no room to fit it in, and it's not going to be able to initiate transcription. So you stop dead in your tracks right there. But as soon as you get that repressor out of the way, then now you're able to move forward, okay? Okay. What about eukaryotes? Okay. Well, yeah, eukaryotes, it's going to have to be a little bit more complicated, right? Because they're eukaryotes. It's also more fun. So you have, remember in your transcription initiation complex and those distal elements of regulation, there were areas where you can interact with activators and repressors. Remember those? Yeah? Okay. So where do they fit in? Well, they're going to fit in to those regulatory spots. Okay, so we have many different genes. We talked about how you could splice differently the same transcript to get different proteins. And we already have a very large DNA. So how are we going to make sure that you can control which gene becomes expressed at what time, in what cell type, and for how long, without having to multiply, without having to have one gene for each of those different situations? Well, this is where activators and repressors come in. You already have the sequence, the genes laid down, but they're the same in all of the cell types. So you don't really need them to always be expressed in all of the cell types. So how are you going to organize that? Well, by having different combinations of activators and repressors, you're able to elicit many different types of responses without having to multiply the number. So instead of having one protein for each of the genes and each of the situations when you want that gene to be turned on or off, you can have a fixed amount of these activators and repressors, but the combination of how they all become part of the transcription initiation complex will allow you to have a large number of triggers without having to multiply the number of these actual activators and repressors. Okay? So they've kind of simplified it into a little cartoon here where you have two different genes. And you don't want them to be transcribed at the same time. But they're under the control, or their promoters are connected. So how are you going to make sure that you don't activate transcription at the wrong time? Well, that's where the activators and the repressors form different combinations. Okay? So for gene A, you would need a certain pattern of activators that will, within the transcription initiation complex, form the proper structure that lets RNA polymerase to start transcription. Without that particular pattern of activators within the complex, it doesn't get started. So that gene would be silenced. Okay? The other gene, you need a completely different pattern of activators. And this is true for all of the genes. Okay? So you'll need a specific pattern of activators and repressors to be part of the initiation complex, but that allows you to exercise a lot of control on which gene is expressed at what time and for how long. Okay? So this is regulation that occurs before transcription is initiated. Okay? Then you go on to transcribe. We talked about how you could regulate your gene expression after transcription is done, at the stage of maturing your mRNA. What about once you have your transcript or even your mature RNA? Do you think there's anything else that could be done in order to control how that gene will effectively be expressed in the cell? Let me ask you differently. Do all mRNAs lead to protein? Once you've transcribed into an mRNA, does this mean you will necessarily have a functional protein? You could form other things from mRNA, like what? No. They won't be formed from mRNA. Lipids aren't from mRNA. You had a...
I would say no sometimes, like if it's an error or something, it will be degraded.
Okay. So in the case of an error, you want to have a fail-safe mechanism. You don't want to get that mRNA to be translated. Good point. What if there is no mistake? Do we need them all to be translated? Or is there anything else going on in the cell? Yep. tRNA for ribosomes. True. So if you are producing transfer RNA, then those won't lead to protein as well. Okay? What about mRNA? If you have messenger RNA, you've transcribed it, it's messenger RNA, you've matured it. Is it automatic? Are they all going to make protein?
Well, probably until it's degraded and it goes into the RNA, then it won't be translated.
Okay, so if that mRNA gets degraded, it's not going to go on to be translated. Good point. So how do you control for that? First of all, what would degrade it? And second, how do you make sure that that doesn't happen? Or is it important to have a way to make it happen in order to prevent translation? Could that be a spot where there's another level of regulation? Where would that mRNA be degraded if we want to stop translation? Nucleus or cytosol? I can't hear you. I'm old and deaf. Nucleus. Yeah, that's where the nucleases are. Okay, so we want this to happen before it gets out. Okay, makes sense. Do we want to be able to control if that happens or not? Or do we just, let it happen? Yeah, you're going to want to exercise some control on that. Okay? And this is where small interfering RNA come into play. Okay? Their job in life is to interfere in transcription to translation. That's what they do. Okay? The cell produces these small portions of RNA, and their job is to interrupt. They're interfering RNA. So what do? They go and they target different mRNAs, bind to it, and that directs them to be degraded. Okay? So it happens in the nucleus, and it's a good process to have in the cell. If you've not been able to regulate the level of expression, so you've got too much mRNA transcript, this is a way for the cell to make sure, hang on, we don't need that much protein. Okay? So let's get rid of a few of these. And to make sure that you're not relying simply on nucleases recognizing mRNA as being inappropriate, because they are appropriate. They would have gone on to translation. So you can't just rely on degradation. You have to actually target it, direct it, and bring it to degradation. Okay? So that's where these small interfering RNAs come into play. Another regulatory process that occurs in this cell is called microRNAs. Okay? These are a little bit trickier. MicroRNAs are very small sequences. So there's a gene that allows to produce them. Transcription produces these small sequences, and they're pre-mRNA, just like the other mRNAs that we produce. But the sequence is such that it's so complementary, it's going to right away fold over itself and form this particular structure where there's a double strand and a hairpin loop. Okay? That doesn't look like pre-messenger RNA, does it? No. Our pre-messenger RNA is a nice linear strand. So this is a little bit inappropriate. And it doesn't have a 5' or a 3' cap. It doesn't have any of the features that you're looking for in a pre-mRNA transcript when you're a eukaryotic cell. And yet, it's able to pass through the nucleoporins. So now it's in the cytosol. Okay? What's going to happen here? Well, it's going to be met by a protein, an enzyme actually, called dicer. What do you think it does? It slices. Okay? And it's going to dice this small microRNA, and it's going to get rid of that hairpin loop. Okay? So now you've gotten rid of that little hairpin loop, and what are you left with? A very short double-strand segment. Okay, complementary. That looks like a very small portion of double-stranded RNA, or it could pass as a very small segment of DNA, right? It's double-strand, it's nucleotides. Okay, then what? Well, we have this large protein complex that recognizes that and binds to it. Okay? And when it does, they form this large complex. And that complex is going to say, wait a second, RNA shouldn't be double-stranded. It's single-stranded. Oh, let's get rid of one of them. And now you're left with a very short single strand of RNA, which is why we call them microRNA. But this strand is effective. There's no issues with it. It's a proper sequence of RNA. Okay? And that protein complex has enzymatic activity. And now what it's going to do is it's going to look for mRNAs, mature mRNAs that have passed the nucleoporins out of the nucleus into the cytosol on their way to finding a ribosome for translation. But wait, they're going to grab them. They're going to grab them, and now this prevents translation from occurring. Okay? So how does it do? Well, the pairing could be imperfect. Okay? So just enough recognition between that microRNA that's within the protein complex and the messenger RNA that's been matured and let out of the nucleus. There's going to be some recognition, some pairing. That pairing might not be perfect. If it's not perfect, all that's going to happen now is prevent that mRNA from becoming or meeting a ribosome. So you essentially block translation. And that will be blocked until that pairing remains. The mRNA isn't affected. It's not degraded. It's not deteriorated. It's just held back. Okay? And then eventually that interaction can dissociate and the mRNA can go on to be translated. But if that pairing was absolutely perfect, no mismatch, exactly the right pairing, then right away that messenger RNA is going to be degraded. Okay? So even in that event, there's a little bit of a fail-safe switch. You can just delay translation, or you can prevent it from happening because that mRNA has been destroyed. Okay? Yes?
I don't understand why it destroys it if the pairing is good.
If the pairing is perfect, then that's what triggers the degradation. Okay? If it's imperfect, it means, you know, there's something going on. I'm recognizing you, but I'm not recognizing you entirely, so I'm just going to hang on and see what happens. So it delays translation. Until they're separated, translation can't occur. But if the matching is perfect, then there is no uncertainty. You match my sequence, I'm going to destroy you. Because it's a perfect match. Yeah. Okay? All set? Questions? Yeah. Is the, okay, wait up.
Is the protein complex like the purple one?
Yes.
When it's perfectly matched,
Yeah.
will the whole piece be degraded or just the strands?
Oh, just the messenger RNA that's been recruited. The microRNA portion, part of the complex, remains intact. So it can go on and grab another one. Okay? Yeah.
What happens to the other piece of microRNA that was like ditched?
The one that was ditched, it gets degraded. That one's done. Okay? So it was there just to be able to pass through and make sure that you can initiate that complex. Once that's done, bye-bye. No more. Yes?
Is there like a specific number of or length of the sequence that needs to be different for it to realize that they're not the same and for it to let it go, or it's just kind of like...
That's a good question. I don't have an answer to that. The question was, is there a specific number of sequence that has to be imperfect or mismatched? I don't have an answer for you. I don't know. And this is a fairly new process as well that we're discovering. But it's a really good question. Let me look into it. I might have an answer for you. Okay? Yes?
So how is it imperfectly paired?
So the sequence, the nucleotide sequence on the microRNA that's part of the complex and the mature messenger RNA that left the nucleus, those sequences can pair up. A, T, G, C, G, C, T, blah, blah, blah. Well, U's because they're RNAs, sorry. If the matching is absolutely perfect, then you just hold on to it. If there is any mismatch, and that's Eyal's question, how much mismatch do you need? I'm going to look into that. When there's mismatch, then you just degrade it. Okay? All set? Okay. Which brings us to another little piece of DNA information called transposons. Who's heard of transposons before? Okay. So they're essentially jumping genes. What does that mean? There's a little portion of DNA that can change places within the genome, right? We talked about chromosomes. We talked about genes having an address, that you know exactly at which position on the chromosome you should be able to find a specific gene. Remember that? Okay. Well, transposons are portions of DNA. Small genes, small portions of DNA, not necessarily entire genes, and they can change places. They jump from one area of the genome to another. Okay? In humans, for example, these jumping genes, these transposons, they represent half of your genome. Half of the DNA in each of your cells is sequences that can change places throughout the cell's life. Okay? This was discovered when Barbara McClintock was working on corn, or maize, and she discovered these sequences that were changing places. And it was part of how different phenotypes were being observed in the corn. Okay? So how does it happen? How can you have one portion of DNA jump out and come back and insert itself elsewhere in the genome? How do you think that happens? How is that possible? You're taking both strands, just one strand? How are you going to do it? Right? Okay, so let's talk about that. So how do they move? Okay, well, if it needs to be removed from the sequence, something needs to cut it out, right? So we talked about different enzymes when we were removing introns. Okay? So now we're on DNA. We're not on RNA. So we can't use the spliceosome or those enzymes. But there are other enzymes in the nucleus that can do similar tasks. Okay? And they're called transposases. Okay? So they're enzymes that recognize specific sequences. Okay? And how they do that, we're not entirely all sure exactly what the mechanism is yet. But, you know, it's being studied extensively, and it's very interesting because you're going to see what the impact can be. So this enzyme is able to cut certain portions of DNA. But then you need to be able to stick them back in. So how are you going to do that? Well, we'll see. But more importantly, where? Where do you stick them back in? What happens if they are inserted into a non-coding area? They're useless, like it has no impact, right? So you've got this little portion that ends up being part of a non-coding area. That's not really going to have any effect on the cell's activity, on the cell's machinery. But what if it lands smack dab within the gene for myosin? no myosin. Yeah? Your myosin, the transcription of that gene is going to be affected. There's that sequence of transposon that's in the middle of the coding region. You're not going to get intact myosin at the end, right? So that's why it's important to understand how these portions work. Now I'm making it sound like they're all bad, but they're not all bad. They're not all bad. They're actually part of how you can shuffle all of your DNA. What do I mean by shuffling? What do I mean by changing the sequence of your DNA and that being a good thing?
You can have short left DNA but still have the same variety number of nucleotide sequences.
I'd go one up on that. Instead of saying same variety, you're introducing more variety, right? Okay, what's the basis of evolution? How does a species evolve? Keeps the best traits. What else? Mutations. What else? How many of you have done 1130? Okay. How does a species evolve? Genetic variability. Diversity. Well, you have all of these transposons running around, changing places, differently between you and you and you and you. Oh, look at that diversity happening right before our very eyes. Okay? So this is why we're very interested in understanding how these things work and what their role is and also what the result may be. This is actually part of the mechanism how bacteria acquire resistance to certain antibiotics. Right? Why is it that now we're very sensitive at not prescribing antibiotics for no reason? Right? Because they've become very good at being resistant to all of the series of drugs that we have available. And we're running out. We're running out of available drugs against bacteria because they've acquired resistance. They've acquired the ability to ignore those drugs. Okay? How do they do that? Transposons. They obtain and they change the sequence. Okay? So the actual sequence, if it lands inside a functional gene, it has a negative impact. But if it lands elsewhere, then that impact can actually be quite beneficial for cells. Okay? One example in humans, the ALU transposon. Okay? That represents about 15% of each and every one of your genomes. And it's not a very long sequence. It's about 300 base pairs. Okay? So it's shorter than a gene. Yeah?
You said if it lands in the middle, it would be harmful, but anywhere else it would be beneficial.
Well, if it's in a non-coding region, then it's introducing variability. It might bring you new traits, new abilities. As in,
Wouldn't you not code that area because isn't that going to get cut out?
it depends where it gets cut out. Okay? Here's an example here. Okay? That sequence has landed between two introns. So it's within a non-coding region. Okay? What happens to it? Well, there can be splicing that occurs, recognizing these introns. The two exons match, and that transposon is removed. So it has no impact. It was part of the DNA sequence. It was transcribed as part of the pre-messenger RNA, and it was spliced out. So no impact. But what if you had alternative splicing, and it became it acts as a new exon. So that means that it was transcribed, it was retained during splicing. So that messenger RNA will be mature, but it's quite different from this one. Do you agree? Okay. So what's the result for translation? Well, now that portion will be part of the translation. What's the impact? I don't know. Without the protein sequence, I don't know what the impact is. But you might have just given that protein ability to increase affinity to a ligand, interact with a whole new family of ligands. You might have given it the ability to be expressed in a different cell type. So those changes may or may not be bad for the cell. We don't know until we know where it lands, actually. Okay? But we do know that for certain of these ALU transposons, it is bad. They're associated with quite a few diseases, some forms of cancer, Alzheimer's, and hemophilia. Okay? So the idea is that they're not necessarily bad. They're not necessarily good either. It depends where they land, and it depends how splicing and how their expression will be regulated. Okay? Any questions on what we've covered today? Okay. So next lecture, we're going to pick up right here, and we're just about to get into translation. Okay? So we're finally going to see our protein. Have a great day. Oh, and Thursday, we're going to do course evaluations. Okay? So we're going to do that at the beginning of the lecture.